Good afternoon, everyone. My name is Seyifunmi Michael. I'm a PhD student in University of Cincinnati from the Meraldi Lab. And today we're going to be talking about gene regulatory network inference optimization with mammalian gold standard datasets. So in our lab, one of our focus is understanding cell behavior in vivo. And basically, we are interested in as we know, cells are constantly carrying out chains of activities ranging from metabolism to external response to stimuli. The important question is, what are these activities and are these cells doing it? To answer these questions, we need to understand the regulatory mechanism that governs gene expression. And gene regulatory network inference provides us with a framework for this, uncovering which transcription factors are in charge and how they orchestrate gene expression. Understanding this network is key to figuring out how genes are turned on or off in different disease concepts and in also identifying critical TFs and pathways that drive disease. However, accurately inferring GRN is still a big computational challenge. One of these challenges includes high dimensionality. There are many model parameters to learn. Oftentimes, we have potential TF gene interactions as many as 1 million or greater. Then we usually do not have enough variation to tease out TF gene interactions. And also, it's difficult to separate real interactions from noisy or spurious correlation. So the core challenge is, or the core question is, how do we narrow down the possibilities and recover the true underlying regulatory architecture? One way to improve GRN inference is to incorporate other sources of information. A commonly used source is to leverage CF binding information derived from ATAC-seq data. And ATAC-seq data identifies open chromatin region and also gives us information about potential TF binding sites. Combining ATAC-seq with motif scan helps us construct an initial noisy prior, which we could use to refine our GRN and also use in GRN modeling. These are just some examples of studies where TF binding predictions have helped improve GRN inference, like Scenic Plus, Network CF, there's Superfactor, and so many other ones. For our method or model, we adapted a previously published model called the inflator, which models gene expression as a linear combination of TF activities. It takes as input gene expression data, which could be on a single cell resolution or a pseudobulk resolution, along with a prior TF interaction, which I talked about earlier. We derived this from our tag data. We also estimate TF activities in two ways. One way is from our initial noisy prior of TF target genes, or we use the TF expression as a proxy for our initial noisy prior. So now, our model also uses adaptive lasso for feature selection. We need to select important TF gene interactions. And unlike the standard lasso, which penalizes all interactions equally, we use an adaptive lasso, which gives smaller penalties to TF gene interactions that are present in our prior. And this helps prioritize known interactions while also allowing for discovery of new interactions from our data sets. Having said all this, I'm going to introduce our data sets, which we used in this benchmark. We made use of two benchmark data sets. One is a single cell RNA-seq and single cell ATAC-seq data from in vivo splenic CD4 T cells from young and old mice. This data set includes 16 CD4 T plus cell populations. We also used, for benchmarking, we created a gold standard network of TF gene interactions in CD4 T cells. This was based on TF ChIP-seq and perturbation experiments followed by RNA. The network includes a total of 50 TFs, and it was created by our postdoc, Joe Wayman. And this network will be used in evaluating our GRN for the mouse data. Our second data for benchmark is a single nucleus multi-omic data set of 14 CD4 plus population from the blood of 4 human donors. Then I have a heat map here just showing the transcriptomic profile of this cell population. And one thing we see here is we see a stronger activation dynamics in the memory population compared to the naive population, which is the first one here. We also generated a gold standard network from 3 databases. For the TF ChIP-seq data gold standard, which was uncreated, we generated that from the ENCODE database and the SystemDB database. And this was done by our 4th-year PhD student, Alex Katko. And then we used the TF perturbation dataset from this paper, Winstock.js, as our knockout gold standard. And this Venn diagram just shows the number of interactions within each gold standard. And then the number of genes when we combine both gold standard, which the ChIP plus knockout happens to be our most trusted gold standard. Then for both the in vivo CD4 and dynamic CD4 human datasets, we constructed gene regulatory networks using both the single cell and pseudobulk gene expression data. We then compared our performance with GRN from single cell RNA and single cell ATAC-seq data. We also evaluated our performance based on how well the networks we generated were able to recover TF gene interactions present in the TF ChIP-seq or knockout gold standard. So for the in vivo CD4T datasets, we see that networks built based on our single cell data outperforms networks built from the pseudobulk data, except when evaluated on the ChIP gold standard, which is the first image over there. Which highlights an important point from what we see here. We see that performance of our gold standard can vary depending on which gold standard is used for evaluation. From the ChIP, we see pseudobulk performing better, and the others, we see the single cell method being better. Likewise, on the dynamic CD4 dataset, we see the single cell derived networks consistently outperforming the pseudobulk methods in all of the 3 gold standards. We also compared the performance of our method with other state-of-the-art methods, Cynic Plus, which is based on single cell. Celeraco also is based on single cell. And Superfactor is a deep learning model that takes in your pseudobulk data and a prior. And one thing we see here for the in vivo CD4 dataset is that on the ChIP gold standard, we see that Celeraco outperforms all other methods, but then it has a very low recall. The recall is not up to 0.05. And on the knockout gold standard, our infralator models, both the pseudobulk and the single cell, performs better than the other methods. On our most trusted gold standard, which integrates binding and functional evidence, that's the ChIP plus knockout with 14 TFs, performs comparably to the networks, because when we look at it, the difference is not really much. It's not even up to 1%. So then on the dynamic CD4T datasets, we see that the infralator networks from our single cell seems to have better performance, except for the ChIP gold standard, where the Celeraco performs better, which is something we also see in the in vivo mouse dataset. But then the Celeraco also has a lower recall as seen in the in vivo dataset. This also reiterates the point that the performance of your network also depends on the gold standard datasets used, because there are many models or methods, benchmark methods like Beeline, that makes use of ChIP-seq, and they showed that they have high performance, but this is showing that it's dependent on what gold standard you make use of. We're also interested in one question, which is how does our single cell derived networks, how does it scale? And at what point do we see diminishing returns in performance when we increase the number of cells used? So to do this, we did what we call a downsampling, where we performed a stratified random sample based on our cell types to generate smaller data sets. So for the in vivo data sets, we generated higher resolution data sets with 1K cells, 10K cells, and 30,000 cells, and compared performance. One interesting thing we see here is that on the cheap gold standard, which is a little bit counterintuitive, we see networks built with lower number of cells having better performance in the cheap gold standard. While on the knockout gold standard, we see that as we increase the number of cells, we see better performance in our networks based on precision recall. And for the cheap, the difference is not that great, especially when you move towards higher recall. And also, when we perform the same evaluation on our dynamic CD4 datasets, we see that as the number of cells increases, we tend to see better performance. But this is not the case in the cheap plus knockout gold standard. As we can see here, this 63K seems to be performing worse compared to 10,000, especially from recall between 0 and 0.5, and then seems to pick up, and then they have almost similar performance. So the key takeaway from our work so far is the generation of two data sets, the in vivo mouse and the dynamic human data sets. We also generated gold standards based on TF binding and also functional TF perturbation. And we've shown that single cell outperforms the pseudobug-derived method, especially when you have at least 10,000 cells. And we've also shown that single cell method performs better than some state-of-the-art methods. Then one key takeaway also is that gold standard performance evaluation depends on the underlying data. Are you evaluating your network based on TF ChIP-seq or based on functional gold standard? We also see that the dimensionality of your gene expression data has a lot to say about the performance of your network. Then one of the things we're working on right now, or we're going to work on in the future, is right now we're only using one source of prior information, which is from our attack data, to build our networks. So what happens when we combine several sources of prior? Say we take in a max attack model, CF information, which is one of the models in our lab also. It's a deep learning model to predict TF binding sites. Or what if we use a compendium of database like the collectory as our prior, and what happens when we integrate this with other sources? Does it improve performance or not? We are also interested in building cell type specific GRNs, and then see if when we combine this cell type specific GRN with our single unified GRN networks, which we've built here, are we able to improve performance? We also want to build a multitask learning approach, which borrows information from a particular cell type GRN and uses those shared information in other cell types to improve the performance of our model. Also, we want to build a middle ground between using pseudobug data and single cell in building our networks by using what we call a meta cells. There have been papers on meta cells like the C cells and meta cell paper. So basically, that's what we're working on right now. And these are my lab members. That's my advisor in the middle. And I would like to thank everyone for listening and also thank my lab members. So do you have any questions?
Thank you. Great talk. So you have the benchmark data set, so you have the gold standard or ground truth. I noticed that in your precision recall curves, the recall is very low. Like your predictions, you're only predicting about 5% of the methods. Did you look at the remaining true positives or the remaining positives in your gold standard data set, did you look at how the model scored them, why the model excluded them? Is this like because of correlation is low or some other reason?
Oh, yeah, I see what you're saying. So basically, for some of our networks, we have longer recall, but then I chose to show specifically for the infralator models, not the Cynic Plus and the other models. For some of them, we have longer recalls, but I just decided to show just the 10% recall because based on how recall is calculated, you rank things based on confidence and stability. And we believe that networks within, say, 10%, 20% recall, I mean, TF gene interaction within that range will be the most confident um, interactions. But then, did I look at the correlation? Honestly, no, I did not, because I was only thinking about precision recall to evaluate. But then, it's something we could look at.
Okay, thank you.
Yeah, thank you.